sugar gliders.
Sugar gliders. All right. I like sugar gliders. Tell me about sugar gliders.
I was like, I had no idea that anyone is doing CRISPR genetic engineering in marsupials and trying to do anything with them. And so I thought that could be a very exciting place to go. So the sugar gliders, y'all, these animals are marsupials. They have this membrane from their hands up to their legs, or down to their legs, and they can fly. Well, glide. And the question is, Cricket, why can these animals glide when other marsupials that are close relatives cannot? Right, Cricket? That's a great question. Why can these glide when others cannot glide? These animals can fly. There are some that cannot, and they're looking at the phylogenetic tree. So what they did is they did a bunch of genetic sequencing, and they found that there are differences in the animals that can fly, can glide, versus the animals that cannot glide. And they identified what they thought was a gene that is turned on at different rates in the development of the animals that can fly versus the ones that cannot. And so that's what they were trying to study, is what are those differences on a molecular level. So one thing they point out, y'all, is that's really cool about this experiment is the sugar glider is a marsupial. So the babies develop inside their pouch, and they're really, really tiny when they are born. They're the size of a bean. And when they are born and they move their way into the pouch, they actually don't have those gliding structures developing yet. That comes a little bit later. So what you can do experimentally is put the mom to sleep, take out the joey, do some experiments on it, see the development of the wing, the gliding structure, and then put it back in to the pouch. You can't do that with a regular mammal, right? You can't open up the mom, take out the embryo, take a look at it, do stuff with it, and put it back and close back up the mom and everyone be hunky-dory. You just can't do that. And so this is a very unique system. So now he's going to go into some of the experiments that they did, and then we'll look at the paper too to further demonstrate that. So we're just going to go into the paper, nothing too crazy in terms of all the different components, because there's a lot going on here. There's a lot that went quite right, and then there's a lot of very strange things that they're also doing in this study, but we're going to jump into some of the beginning parts. And here, just right off the bat, they're looking at pup embryo imaging. So again, this is the part of the sugar glider that you're trying to look at and figure out what are the genetics behind this. Why do they form this membrane that allows the animals to glide while other marsupials don't have this? And so you can actually see this is the pup at day 5, day 8, and day 12. By day 12, you already have this membrane coming in. And like they said, what's really cool is you can actually pull the animal out of the pouch, take a look at it, image it, and put it back in the pouch, right? That is not something you can do for developmental biology and other mammals in general, like mice and rats. Once you take it out, you can't put it back in. And so this is a very unique system to be able to study this. I hope that it'll expand the door to more marsupial studies. They even identified the group of cells that lead to this. So it is right here, this primordium segment of the animal, of the embryo in this model that they have, that's where the cells are coming from. And with this, they also actually they did a ton of different types of analysis. So actually, for those wondering earlier tonight when we were talking about accessibility in science and the price of things, here they did 2 kinds of sequencing that they're demonstrating here. They did ChIP-seq and they did ATAC-seq. Now those are 2 different kinds of sequencing experiments. From that, they got 68,000 candidates from ATAC-seq that are different in these flying, gliding animals versus non-gliding. They got 127,000 plus candidates for ChIP-seq, and they looked at the overlap to figure out exactly what candidate genes they should be analyzing. This panel right here, just panel D alone, you're not seeing the whole results of the data. That's in the supplement. That is easily $50,000, right? And this, remember, is kind of getting to what we talked about at the very beginning of the stream. This is a Nature paper. This is one of the biggest level paper that you can get, and a $50,000 experiment is in panel D and E, right? Sam, that's a little bit shocking. You would predict, or I would maybe naively predict at the beginning of me looking at these kinds of things, is that level of experimentation and that price would probably go elsewhere. It would how it's being driven and where it ends up. Like, it's would be a much bigger element to it, but they just used it honestly as a means to get to a candidate gene. And that it used to be like maybe 15 years ago, if you did an experiment like that, that experiment alone would be enough to get you a really high tier paper. Now in these bigger tier journals, a lot of these studies, as they integrate it in, all these really end up doing is just demonstrating like, hey, this is a small segment of the study, it got us a candidate, and we moved on. And so that's just, I think, a nice little illustration of how some of these things work. Further down, we're going to skip, here is just some of the more sequencing results that they did to look at how chromatin's interacting. I'm going to get to the imagery component of it. So this right here is the joey, and the joey's totally fine here. They take the pouch, they can essentially loop the pouch out so they can get to the joey, and they identified where they needed to do injections on the joey. The joey, remember here, maybe no one else has this problem. I did. It is not a it is not the kangaroo. It is just a broad term for our marsupial friends. So for the sugar glider, its baby, who's inside of the pouch, is called the joey. Just to give you all that little bit of extra detail. So they inject in this site where they identified the patch of cells that make this membrane. They inject a lentivirus, and that is going to mark certain tissues and certain cell types. So here you can see the bright field image. You can already see this is 10-day post-birth. You have right here that membrane. And then here at the injection site, what you're looking for is the GFP, that glowing green, and that's your cell population that you're interested in. This glowing part right here. The rest of this glow is called autofluorescence. Just under a certain wavelength of light, things glow. And that's not true glow. It's whatever is above that background that is actual glowing that you have. And so what you're seeing here is that really, really bright part is where the injection site is. Yes, yep, exactly, Gers. I was not aware of this, Gers, while reading the paper. And so as I was reading the paper, they kept referring to it as joeys. And I'm like, wait a minute, did they switch to kangaroos? And then I looked it up, and yes, you are correct. It is for all baby marsupials as they live in the pouch. I apparently, Gers, have been living under a rock, and I thought it was just for the kangaroos. Probably because that's the most common location where I hear the description of it. But yeah, so that's the injection site. They're able to mark all the cells. So in that image, the green are the cells that they hit, the blue are nuclei. This is a different flavor and image of cross-section. This is an actual cross-sectioned sample that they're looking at, and what they're trying to do here, chat, is to get rid of that membrane with genetic tricks. So here, you're comparing the uninjected side with the injected side. And this image here is your sham injected. So you inject everything into that segment of tissue, including the genetic thing that is used to turn off a gene. It's a genetic tool. But Gers, there is a genetic tool called the scrambled RNA. It's essentially a molecule that does not bind to any gene, but it's same fundamentally as something that does bind to and turn off a gene. So it is the most appropriate control to have, because you can compare this with what's going to be below, and if you see, no difference, that's the most appropriate comparison. You wouldn't compare the uninjected one to injected only. You'd want to compare the scrambled or sham injected to the experimental, because then, there's always going to be the counter argument, well, what if you're only seeing an effect because there was something that was part of the injection process that caused there to be a difference, and that's the only reason why you're seeing a difference, and not because of what gene you turned off. And so that's an important element to make sure to account for. So remember, y'all, this is the normal injected with nothing special in it, should do nothing. And you see there's the membrane there and the membrane on the other side. So the animal still has that membrane forming. It's still going to be able to fly. What happens if we turn off the gene that was identified in their screen? Look at that. It shrinks. That tissue goes, and so all by turning off a single gene. And remember, what this is a small RNA injection. What this means is it is not genetic engineering. So in the rest of the body, there is still normal levels of that gene. It's only where they injected that gene is turned down. It's not even turned all the way off because of just the technology. It can't turn everything all the way off, but it can turn everything mostly really down. So it's like that dial switch. Instead of having setting on 11 like in your Tesla, you might have it down to a setting of a 2. So why that's important to note is sometimes if you change from an 11 to 2, doesn't have an effect because you need so little of the product to have your final output that there's not going to be any kinds of change. That is not the case here. Moving from that 11 to 2 does in fact cause this structure here to shrivel down. And so we'll see another image, but basically that membrane from the arm to the leg gets pulled right on back. And so they're identifying a gene that's actually part of the development and therefore with the earlier part of the study, the evolution of flight in these marsupials. And then here's another different kind of cross section that you can compare here. So this right here is marking the protein. So, right, you might be wondering, sure, you've done this injection, but is there an actual output difference? And in fact, there is. So here is your uninjected membrane, and you see all the red. All that red is this gene product, this protein called EMX2. And this is your normal membrane. There's red all over the place. This is your injected where you've turned down the levels of EMX2. Notice two things. The membrane is shriveled, so it's much smaller. The scale bar is the exact same between the two images, which is very nicely done. It's smaller and shriveled back, and there's a lot less red. And so that's like the really like hitting home the idea that yes, they are in fact doing what they say they are doing. So it's another way of validating what they did. And so Sleazy, great question. That is not gram staining. This right here is marking this protein product EMX2. So everywhere there's red, there's protein EMX2. This is your normal membrane, and this is where you've turned down the dial of how much gene product there is. So instead of, again, let's say you have a gene level of 10, you've turned it down. You can't turn it down to 0 using this technique, but you can turn it down to like 2. And so you would predict to see less red, and you see less red. The other thing too, Sleazy, you see it's smaller, and the reason it's smaller is because that protein is what forms the membrane for flight. But yeah, so the yellow is the upregulated genes that have gone up when you do the comparison, and the pink are the genes that have gone down. And so actually what's really cool is, for all of our geneticists who are in the audience, some of these genes are developmental genes that we know are involved in cell growth. Wnt, HOXB, there's EMX2, PAX1. Those are genes that we know are involved in cell proliferation, which makes sense, right? If you are growing this membrane from your hand to your leg to allow you to fly, you need cells to divide. And so if you're getting rid of genetic elements that contribute to cells dividing, it makes sense that those are the genes turning down. All right, so the rest of the paper, they had 59. So this is that genetic sequencing that we talked about. So they did genetic sequencing, right? They tried turning off this gene, this EMX2, and actually it's really nice that when they turn off the gene EMX2, it is in the turned down genes in the sequencing. This is a nice validation that everything is working right. So they turned down the genes, and they compared what's normally up versus what's down, and they found 59 candidates that overlap. So that's what they're looking at now. Of those 59 genes, what's important. They were looking, remember, at this gene EMX2, and this gene EMX2 was forming the connection point, the membrane between the top of the animal's arm all the way back down to its leg. And so they were able to show in this earlier figure that if you turn off this gene, right, the membrane shrinks. So instead of having this nice regular size membrane at this control injected, at the experimental one, it shrinks down. So you've demonstrated here one element of genetics that's critical. What that means, and what the element of that is that for a gene to be truly involved in a phenomenon, you have to demonstrate that it is necessary and that it is sufficient. So necessity here means if you turn off the gene, does the thing that you're looking at go away? Here, they were looking at a membrane, and when they turned off this gene, the membrane receded. So, okay, yes, there is something here that is in fact working. Now they have to demonstrate that it is sufficient. What that means is they need to turn on this gene in something that normally does not have it on and see if they can recapitulate or remake what they observed with the phenotype. So, can you give that membrane, gliding-like membrane, to something that does not have it? Probably the best experiment would have been to use one of those non-gliding marsupials and try to give the non-gliding marsupial the ability to glide. However, they went with a tried and true, probably easier model to genetically manipulate, which is a mouse. So what they did was in a mouse, they injected this EMX2 from your sugar gliders, that gene and this genetic element. And what they wanted to see was, okay, can we turn it on? Can we make more of it? And will there be a flap that forms? So here, you're seeing a developmental stage of a mouse. What we're marking here in purple is EMX2 levels. And so they need to turn on EMX2 levels in this area here. And that's what you're seeing. See where the arrowheads are? That marked banding pattern, that's the area where they're wanting to inject and that's where they did inject to increase the levels of EMX2 to see if you can get a gliding ability in these animals. And then, humans would be too big, unfortunately. So when you do the comparison, right, so there's the control here, and here is this transgenic. What that means is you've taken the gene from your sugar glider and you've put it into the mouse. And what you see here, I think it's pretty striking, is that there are more dots in the black and white image. So these two images are at the same magnification, and then both of them have a little square that they zoom in on. And what you're looking for are these dots. These dots are what are marked by a molecule called DAPI. That marks nuclei. That marks DNA. So each one of those dots is a cell. And so what you need to take away from this is there are more cells, right? I think it's pretty, if I can see it like this, I don't even need the bar graph. Like, the bar graph is important, you need to quantify it. But I really like in this paper, they just put the image, and you can see there's definitely more dots. Now the catch is, this did not form that membrane. They were not able to fly. These mice did not have this giant membrane like the sugar flyer, right? They did not have the sugar glider equivalent. But there's a lot more cells. So it's the starting point. They had some kind of membrane form. It wasn't the full one, it wasn't even half of one, but at least the beginning stages of it. And what the researchers suggested is, you have all the extra cells, but they're all clustered together. And they need to be more spread out like your sugar glider. And so probably there's other genetic elements that are important to help push those cells further out to distribute to make the membrane. That is the hypothesis. We'll look at another image in just a second. We have a question from Glued with Gold. Glued, I am so sorry I missed that question earlier. Thank you so much for reposting it. Is it possible that sugar gliders will eventually evolve to have actual flight? So that is a great question, Glued, and I think I have a multi-pronged answer for you, which may or may not be satisfactory. So their body type is one that is almost built for gliding. So our bats, we know, have the ability to fly, but they don't have this membrane that's in between their two limbs. They've got an outstretched arm that has its own independent membranous characteristic. And yeah, there's some membrane that's connected down to their legs, but they're primarily having this ability to have uplift that's further away and independent from their bodies. The sugar glider doesn't have that. It just has that membrane from its arm straight to its leg, and it's just that region. That region, at least based on the models that have been done, doesn't allow for them to flap and gain lift. And so that's the big catch there, is that right now, their physiology does not allow for that to happen. Now, Glued with Gold, it's one of those things, never say never. There could be an evolutionary push, some kind of stressor that maybe selects for a mutant that has a little bit more of a membrane coming further out, right? Remember, with bats, it's their hands that has the membrane, so their fingers are massively extended and there's membrane between them, and they're basically just clawing in the air at air, and the viscosity of the air allows them to stay up. That is very, very different, right, than how the sugar glider flies. And so you'd have to either push it towards being more similar to a bat, which would be an absurd amount of evolutionary time, or you'd have to probably change bone density, probably more of the structure of the membrane and the location of it as well, for it to be successful. That's true, Sam, but it's also one of those things that if it ain't broke, right, and there's nothing pushing it to fix it, then it's not gonna be fixed, right? So if there's no evolutionary selection pressure, presumably on the lizards too, that they need to fly to survive, right? It's kind of sad, right? But you take the love out of this, right? These animals, their sole purpose is to pass on their genetic information. And as long as they can do that and there's nothing preventing that from happening, your natural selection really isn't going to push it in that direction for full flight. Makes sense. I was just thinking about the doc I watched with lizards that evolved to glide, prehistoric, that eventually led to birds. Gotcha. No, Blue to Gold is a great question. I just think, it's not that it can't happen. It's just that you need something pushing it towards that direction happening. Like in the sense that it needs to be some kind of reproductive struggle, something like that that's pushing it, Something like that kind of push. And then, maybe too, I think it would be closest to, you'd have to probably do the comparison to bats, where the bat comparison, I think, is important because it's, again, a mammal, right? It's not even that they're that closely related in terms of genetics, but it's maybe something like that and getting some of the extra features. Kind of how Gers said, too, the bone density will have to change. And honestly, that might change their feeding behavior as well, because our bats fly to catch their prey. The sugar gliders, might use a little bit of a glide, but then they're also staying put into a tree and so it'll have to be a lot of changes. Okay, so again, y'all, just as a little reminder, in order to identify if a gene is actually part of something, they need to see if it's necessary and sufficient. It needs to be those two components. Necessary and sufficient. The way they show if it's necessary is if they get rid of the gene, a feature goes away. Sufficiency is when they turn on the gene in an animal that doesn't have the feature they're looking at. So here, that's that membrane that allows flight, and then they get that feature. So the way they demonstrate this, as Sam pointed out, is these bonus dots. So what they actually do is they show these two images. The upper one is your control injected, right? So these are injected with nothing in particular, just all the molecules that you'd have in your experimental without the thing that turns on off any gene. And then these dots are the DAPI staining. Those are staying up. Those are showing DNA, marking DNA. This bottom one down here is when they turn on now the gene. They turn on the gene of interest, right? They're looking at this EMX2. They turn on the sugar baby EMX2 in the mouse, and lo and behold, they have a ton more cells. It did not make the membrane. But it did make a bunch more cells. The reason they suspect it didn't make the membrane is because there's other genes that need to be differentially regulated to push that membrane to actually fully form. It's not a single gene that's important for it. Knock-in. So they knocked in stem cell researcher a gene from a sugar, the gliding marsupial, which is again the sugar. I always forget the name of this. Sugar glider. They put in the sugar glider. The sugar glider gene goes on in. So they knocked in the sugar glider, and as they put in the sugar glider gene, it increased the number of cells here in this site. They already showed necessity in the actual sugar glider. Now they put in sugar glider genes into a mouse embryo, and they made a ton of these extra cells spread, which I think is really cool. So then finally, to round out the paper, they had one more imaging technique. Oh, no, that's actually it. There was some in the supplement, they showed the similar technique. So there was more imaging that they did. They demonstrated that there was this increase in cell population, and that increase in cell population is maybe the formation of the membrane, but they didn't actually end up forming the membrane. They just had the start of it form. And so suspicion is it needs to be more genes. So again, you showed necessity, you did not show sufficiency, but you're leaning on the possibility of it being sufficient one day, which I again thought was really, really cool. So that is your science news story. Number 3, right? So to summarize, researchers were looking at the sugar glider. They wanted to know why it had this gliding-like feature right here. What was that membrane from? They did sequencing experiments. The sequencing that they ended up doing identified across they did flying and non-flying these marsupials. They found this region, this genetic element. They then turned off that genetic element in development of the babies, the joey, and they found that the membrane went away. They then put that genetic element into a mouse and found that there was more cells that appeared. I agree with Gers, though. They probably should have looked at the non-flying marsupials that are close relatives of the sugar glider and see if it worked. It had been maybe more likely to work there. I think the reason they didn't do it is ease. there's much more genetic tools and tricks inside of other animals, and so maybe that's why they didn't end up doing so. That's my hypothesis on that front. And again, I thought it was an amazing paper because they demonstrated a lot of different features, including genetics on these sugar gliders, genetic engineering, evolutionary studies, a lot of sequencing from ATAC-seq to ChIP-seq to RNA sequencing. It was a really, really cool paper, and I hope you all enjoyed it as much as I did.